All right, good morning, everybody. Welcome to the MPG Primer. I hope you had a good 2 weeks off and enjoyed ASHG if you attended. I'm really excited to introduce Ben Finander. He is a fifth-year PhD candidate in Chris Walsh's lab at Boston Children's Hospital. He studies somatic mutations in neurofibromatosis type 1 and prenatal somatic mutations associated with maternal infection. He is also a prolific STEM teacher, which you'll see today, and mentor throughout the whole Boston area. And outside of his amazing research, he also loves to travel and hike, which is great. So Ben, thank you. Ask questions throughout, and yeah.
All right, good morning, everybody. Thanks for coming to this primer. I'm really excited to be here, and thank you for the wonderful introduction. So who here studies germline mutations? Show of hands. Most people. Anyone study somatic mutations? One. Okay, I'm excited. I have some family here. So today, my goal is going to be to tell you how not just germline mutations impact human health, but also somatic mutations. So what I've got up here is a diagram of how many cells can be impacted by a somatic mutation depending on when they happen during development. So there's two main ways that we're going to start out talking about how somatic mutations can have an impact. And one way is if they happen really early on in development. So we start out as a zygote, we have one copy of the genome, we replicate that DNA over and over and over and over again, and we mostly do a very, very good job, but we're not perfect. So if we have a mistake that happens soon after fertilization, before gastrulation, we might introduce a somatic mutation that's present in the brain, in the lungs, and the heart. If, however, it happens later, maybe around the formation of the neural tube, then we'll have a somatic mutation that's just present in the brain. And as we go on through the weeks of development, we might have a somatic mutation that would only impact one hemisphere or part of one hemisphere, or maybe even just a focal region of the brain. So what I'm showing you here is that somatic mutations during development impact more cells the earlier they happen. The other way that somatic mutations can have a big impact, if I point it this way. Oh, I'll just click it. Yeah. Oh, it's not even clicking here either. Let's see if I can click around. If I do that, and then I do this, here we are. Okay, we're good. So I mentioned developmental somatic mutations. The more popular example of somatic mutations is in tumors. And this is really illustrative of the second way that somatic mutations can have a really big impact. So we're familiar with the idea that we could have a tumor initiation mutation in a regular cell that increases its ability to proliferate quickly. And as that massively proliferative cell continues proliferating, it will accumulate additional mutations, some of which are also driver mutations that increase its proliferative capacity further. So what we're seeing in this graph here is that over time on the x-axis, this cell accumulates more mutations, and those mutations that increase its ability to proliferate form larger clones. So we have this idea that in development, somatic mutations can have an impact by happening earlier, and we can also have impactful somatic mutations if they increase the ability of a cell to proliferate. But what about somatic mutations that don't cause cancer, right? We're pretty familiar with this idea that cancer is bad for our health and that a cell proliferating out of control is a concern. But are there other cases where somatic mutations can impact the function of our bodily systems? And to get into how our lab started studying this question, I'm going to give you a little quick history lesson of what our lab has done. So our lab's been around for a while. This is like ye old recipe for a paper in the Walsh lab, right? We're very interested in how the brain is formed and also malformations. So what we classically will do is we'll identify patients who have a specific brain malformation, and then we'll look in the DNA by looking at blood consented to be used from that patient, and we'll look for a mutation that's suspicious, put that mutation either into cells or into a mouse or even a ferret if we need a folded brain to study it, and we'll try to create a mechanistic model. So we've been doing this for a long time, and I'm going to give you just one example to illustrate our point here on a paper that I was actually able to contribute to. So this is polymicrogyria. If we know our Latin roots, we can figure out that this means many small folds. So there's supposed to be a bunch of folds in our brain to increase surface area and contact between the neurons. But what happens in these individuals is there are too many folds. And because there's so many of them, they're all very small. And these individuals have reduced intellectual capacity along with other cognitive symptoms. So we did what we said we would do, and we looked in these individuals, we looked in their blood, and we found some suspicious variants in a gene called TMEM161B. So you look at the family tree, you look at the pedigree, you see who has polymicrogyria, who has these mutations. You see it all is concordant with this idea that variants in this gene might be causing this disease. And then we go and put this mutation into a mouse, and we look at the brain to see if there's anything suspicious. And to skip to the punchline, we found something very interesting. So this is what the apical surface of the developing mouse brain might look like if you take a serial electron micrograph. And you see these antenna-like structures sticking out of all of these cells. And they look like antennas because they pretty much are antennas. These are primary cilia, and their job is to detect morphogens that tell the cell what to develop into. So if there's some kind of gradient, for example, Sonic Hedgehog, this organelle is going to detect that. And when we mutate TMEM161B, those cellular antennae are all kinds of messed up. They're blebbed out here, there's lots of debris floating around, and this is consistent with the idea that this gene and mutations in it are important for how the brain develops. So this is just one example of an example we found of a brain malformation, a patient variant, and a mechanistic model. We've done this many, many times across many different classes of genes. We've looked at genes that regulate neural stem cell proliferation, neuronal migration, differentiation, and even neuronal function. And all using this same pipeline of looking at a brain malformation, sequencing the DNA found in the blood, and then modeling it in the appropriate system. And you could probably guess that it did not always go so smoothly. And that's when the focus of our lab started to evolve a little bit. We thought we could do the same thing with another brain malformation called hemimegalencephaly. And we could play with the Latin roots again and see that what we're talking about here is half of the brain is too big. You see on the right side here, we have our nice cortical folds. On the left side, it's all bulging out. It's pressing up against the skull. This is hemimegalencephaly. And we did the normal thing. We went into the blood of some patients, and we sequenced looking for exonic mutations that might be suspicious, and we didn't find any, at least not in the blood. And we're geneticists, so we understand that genetics are indeed the cause of absolutely everything. So this doesn't make any sense, right? This has to be some kind of genetic cause. So we dug a little bit deeper. And instead of looking at the blood and the DNA in the blood, I should say, we looked at the DNA in the brain. And that's when we started to see something interesting. So this is a very old style plot. This is a Sanger sequencing plot of the part of the gene AKT3, which is in the mTOR pathway, where we found something suspicious. So we see these peaks here indicating that the sequence of this gene is what we expect it to be. But in this particular location, we don't see one peak in this site, we see two, indicating that in the brain, there are some cells with the mutation. This adenine residue is not what we expect to see in the sequence of this gene. So if we look at the blood, this is where we look first. We don't see this mutation. It's just the guanine residue. And in the brain, we have two different clones. 17% of the clones have this adenine residue. The rest of them have the correct guanine residue. So now we have some evidence that it's not a germline mutation associated with hemimegalencephaly. It's a somatic mutation just impacting part of the brain. And this makes sense if you think about this image again, right? When we see hemimegalencephaly, we don't see the whole brain affected. We just see part of it. So in this case, we have a mutation that probably occurred sometime around here during cortical neurogenesis and ended up impacting just parts of the brain. It's now understood that hemimegalencephaly is a disease where if you have a larger mutational load, you have a larger part of the brain affected, I should say. So we have a later mutation. The mutational load is going to be low. The amount of the brain affected is going to be low. And then as you get earlier and earlier mutations in genes like AKT3, you have a larger portion of the brain affected. And it's not just hemimegalencephaly that's like this. This is called Proteus syndrome. It's literally a 1 in a million disease. It's very difficult to diagnose because it's so rare, but it is also a disease caused by somatic mutation, E17K specifically, also in an AKT gene in the mTOR pathway. So you can see here that this individual unfortunately had some of these somatic mutations in some bone, and we see a massive overgrowth of the jaw and even the nose area. But depending on where this mutation happens, these might be in the feet, these might be in the hands, it could be anywhere in the body. But in general, we see that this pattern of somatic mutations impacting health when they happen during development is consistent across a few different examples. So one question you might have is, okay, if I made it all the way through the process of development, right? I am a fully grown adult now. I'm done building my brain. All those neurons are ready to go. Am I safe? The answer is no. So it's not just cancer either, but there are also somatic mutations that can build up in proliferative cells in the body. So what I'm going to tell you about now is clonal hematopoiesis of indeterminate potential, or CHIP mutations. So when we're talking about clonal, what we're saying is that these mutations ultimately become part of a larger group of cells that all harbor the mutation because they're proliferative. We're talking about hematopoiesis, so we're talking about blood stem cells. And then indeterminate potential is the key word here. So these mutations do have an effect on growth, but they're not immediately causing cancer. Their potential for proliferation is indeterminate, right? So we have blood stem cells. We have a nice pool of blood stem cells to regenerate all the blood cells that we need for our body. As aging progresses, we may have the formation of a CHIP mutation shown in purple here, right? Or a non-CHIP mutation shown in brown. And then those cells with the CHIP mutation will become a larger proportion of the cells as time goes on. They have an increased ability to proliferate. They start to outcompete. So this makes sense in a theoretical context, but how important is this, right? How many people have CHIP? And it turns out it's about 10% of the people over age 70 have this in their blood. So this is something that impacts a lot of people. A lot of people have CHIP mutations ready to go right now in their blood. And the question is, what happens? So just to skip to the bottom right, the first thing we think of is cancer. And that does happen more frequently in individuals with CHIP. But it's less than 1% per year. That's not the main outcome of people having CHIP mutations. The more important risk is in heart failure, thrombosis, and accelerated atherosclerosis. Individuals with CHIP have a 40% increase in risk for diagnosis of cardiovascular disease. So how does that happen, right? The thinking is that if we have some kind of mutagenic event leading to a clonal expansion and a CHIP mutation, that those cells with the CHIP mutation, especially when they form a larger clone, are more likely to cause inflammation. And inflammation can be bad for a lot of different reasons, but especially for the function of the heart and for clogging of important arteries. So if you look at individuals with CHIP mutations, not just those with heart failure, but those without any coronary heart disease, we see that the calcification of coronary artery is worse in those individuals with CHIP. So yes, we have increased risk for coronary heart disease, But we also have increased calcification of the coronary artery, even when it doesn't lead to coronary heart disease. So these cells with the CHIP mutation are leading to some kind of process that's increasing risk for heart disease without necessarily causing a cancer. So we're talking about a blood mutation. Blood mutation impacting heart function, I guess, makes some sense, right? But could these mutations also have further reaching effects? And our lab is very interested in the brain, as I brought up before. So we wondered whether CHIP mutations might be associated with a neurological disease associated with aging, Alzheimer's disease. So if we look in the brain, and we specifically look for CHIP mutations, all of these genes here, all of these mutations are CHIP genes. We look at Alzheimer's disease individuals and aged controls, we see that they're more prevalent in Alzheimer's disease. Each one of these columns is an individual, and all these rows are different genes with CHIP mutations. So we see some Alzheimer's individuals have multiple CHIP mutations detected. A lot of them have at least one. And these percentages, when you compare across the different rows, we show that there are more mutations in those with Alzheimer's disease than the controls. They're not unique to Alzheimer's, but they're more common in those who do have Alzheimer's. So this is something that happens in the blood associated with a neurological condition. So how does that make any sense? Isn't the brain supposed to be a privileged space where they only have their own cells and the blood doesn't necessarily get there, right? An important thing to consider here is that Alzheimer's disease is associated with some infiltration of blood-derived macrophages into the brain. And also, there is a similar group of cells to hematopoietic stem cells in terms of microglia in the brain itself. So there's kind of two hypotheses we have for what might be going on here. In the normal brain, we have microglia that are the resident immune cells of the brain. They can randomly mutate. We don't have a CHIP mutations. This would lead to normal aging. But if we do have CHIP mutations, maybe as a result of some inflammatory process, maybe some tau tangles accumulating, or we might have infiltration of blood-derived macrophages with CHIP mutations. Both of those events could lead to a population of cells in the brain with CHIP mutations that could lead to increased inflammation. So there's a couple different possibilities for how CHIP mutations, whether they're in the blood or whether they're in the microglia of the brain, could lead to increased activity of microglia, inflammation, and potentially neurodegeneration. Folks who study Alzheimer's disease know a lot of those genes associated with germline mutations in terms of Alzheimer's disease have to do with immune activity. So this would be consistent with the idea that Alzheimer's disease has to do with immune function. So we've talked about a couple of different ways that somatic mutations can be important. We've talked about ways that germline mutations can be important too. So now I'm going to start to categorize these, and we're going to talk about a third type of mutations and how they can impact human health. So in a germline mutation, all of the cells of the body have it. And for whatever reason, it's more impactful on one part of the body than another. We talked about TMEM161B having this very important impact on brain development. We also talked about examples where the mutation isn't present in every single cell. It happens at some point during development, or maybe it happens after development. But in any case, it's just a group of cells that are related to each other clonally that harbor the somatic mutation. In this case, we see neurons shown with the red somatic mutation and intestinal cells shown with the green somatic mutation. But there's also another possibility. Cells that are done proliferating, they're terminally differentiated, such as neurons in the brain, can still accumulate somatic mutations, right? These we would term private somatic mutations. They're just in individual cells. But, you know, there's like this phrase that people like to say, if a tree falls in a forest and no one is around to hear it, does it make a sound, right? We could similarly ask, if a cell acquires a mutation that doesn't increase its proliferation, does anybody notice? Does anybody care, right? How do we detect that? It's an interesting question, but how do we ultimately detect it when all the mutations I've described to you so far have an effect on proliferation that allows us to notice them. So to address this, Alex Kagan, who now has his own group, did something very clever. So these here on the left are H&E stains of the colorectal crypts. So all of these here are like the villi that stick out into the middle of the intestine and the colon in order to pick up nutrients and water. They're constantly being regenerated and replaced because they suffer a lot of damage. But shown here outlined in these dotted lines are individual crypts. These are just the individual pieces sticking out. And the reason they're special is that each of these crypts derives from just one stem cell. One stem cell out of many that live in this pool here in the inner intestine is going to give rise to one crypt. So that means that if we sequence just one crypt, we can get an idea for the mutations that were in that original one stem cell. So this is going to allow Alex Kagan to ask two important questions. The first is, do individual stem cells in the intestine accumulate mutations over time? Do older individuals have more mutations in their stem cells than young individuals? And the second, which you might be able to guess from the fact that we have 4 different mammals pictured here in terms of these intestinal crypt examples, is whether this age-related accumulation of somatic mutations has to do with lifespan. So in this study, we're going to look at mice, which live 2 years, and humans, which live 80 years. If somatic mutations are just a function of time, then the old humans are going to have by far the most somatic mutations in the study because the humans live the longest, right? But if instead somatic mutations accumulate as a function of lifespan, then a 2-year-old mouse is going to have just as many somatic mutations as an 80-year-old human, meaning that the yearly somatic mutation accumulation rate is going to be much, much higher. And it turns out it's the second option. So here we have on a log scale, we have a mean mutation rate per year. We have mouse here up at 800 per year because they only live 2 years. And then humans way down here at much less, living about 80 years. And all of these different mammals that were also tested that follow the same idea. Increased lifespan means decreased somatic mutation rate per year. And that's, of course, this is all correlative, but that's awfully suspicious. Why go to the effort to increase your DNA repair capacity so much for no reason? Perhaps it has something to do with lifespan, maintaining a relatively high integrity genome in these intestinal stem cells. So one of the things that we can do now, when we have the mutational profile of an individual cell, instead of just talking about one really impactful somatic mutation that increases growth, we're talking about a whole lot of mutations, hundreds of mutations, maybe even thousands of mutations, which allows us to think about where these mutations are happening to give us a sense for mutational processes that might lead to the formation of these mutations. Now that we have so many, we can start to categorize them and look for patterns. And one of the important ways we look for patterns is we look at how each base changes, so which base is changing to which base, and also the genomic context. And the way we look at that is a 96 profile. So what I've got here is the 6 different types of mutations that can happen. We have cytosine to adenine mutations, cytosine to guanine, and so on and so forth. You'll notice that I have cytosine to adenine, but I don't have guanine to thymine. The reason for that is, at least for now, we're only interested in double-stranded mutations. So every C to A mutation is also a G to T mutation, and so on and so forth. So plotted here is the mutation fraction on the x-axis broken down within each of these 6 groups. It's called the trinucleotide context. So it turns out that a lot of the somatic mutational processes that we know understand have unique patterns that have to do not just with the base change, but with the neighboring bases. So we could see here that in this mutational process, this was extracted from the data in that intestinal crypt study that I showed you earlier. We see these 4 towers poking out above all the rest of the mutations that are possible. And in case this is difficult to see, I'm going to give it away to you and say that these are all at CPG sites. So these aren't just C to T mutations in general. They're C to T mutations when that C is right next to a guanine. And folks who study methylation might already know where I'm going with this. It turns out that this is SBS1. This is the result of a somatic mutational process whereby spontaneous deamination of specifically 5-methylcytosine leads to a misrecognition during DNA replication, where that original 5-methylcytosine looks like a thymine and we get a mutation. So a critical thing to note about this somatic mutational process, this signature SBS1, is that this accumulates in fast proliferating cells. Because I mentioned the process, the spontaneous deamination and the misrecognition, that requires DNA replication. If a spontaneous deamination happens in a non-replicative cell, this mutation doesn't occur because you need the DNA replication machinery to make the mistake to even see it at all. By contrast, I'm going to tell you about just one other SBS signature called SBS5. So this signature, we see it's a little bit more balanced across all the different groups. This mutational mechanism is not currently well understood, but we see that it accumulates even in cells that are done dividing. So whatever's happening here, it probably doesn't require DNA replication because it accumulates in cells that are terminally differentiated. So I'm just telling you about SBS1 and SBS5 for now, but in this cosmic database, this catalog of somatic mutations in cancer database, there are many dozens more of these somatic mutation signatures. So if we go back to our intestinal crypts, one thing we can do with this data now is we could extract signatures and see which of these mutational signatures is really driving this age-related accumulation of mutations. And it turns out it's quite clear which is doing most of the work. It's SBS1. These other two signatures did not match as well to specific SBS signatures and were reported as just SBSB and SBSC, but one very clearly matched on to SBS1, and that's the one doing a whole lot of the work in terms of accumulating somatic mutations. And this makes sense, right? Because those intestinal stem cells have to keep dividing, they have to keep making more and more of those crypts as they're destroyed throughout their function. So lots of proliferation, lots of SBS1. So what we can wonder now, we've had improvements in technology. We don't need to be as clever as Alex Kagan and do this thing with the crypts. Now we can study individual cells and just amplify their whole genome and do the same question. Look at individual cells like postmitotic neurons and say, are they also accumulating somatic mutations with age? And as it turns out, they are as well. So each one of these dots is a singular neuron from the prefrontal cortex of a person. And we see that there are more somatic mutations in aged individuals. And these happen despite being postmitotic. All of this time here from 0 to 90, I guess, is not time that the neurons spend dividing, but just time they spend existing. And nonetheless, accumulating somatic mutations. We can also look at disease in this case. We have human samples. We could think about what might impact somatic mutation rates besides age. And here we have two examples of DNA repair deficiencies. This is Cockayne syndrome and xeroderma pigmentosum. And if we normalize for age, even when we normalize for age, the Cockayne syndrome individuals, the XP individuals, both have more somatic mutations than the neurotypical individuals. So we see that DNA damage repair has something to do with accumulation of somatic mutations. We see that when we have diseases where it's impacted, that also impacts somatic mutations. And once again, we can do our signature analysis. So we extract two signatures. This time we find one, signature A, which is a lot like SBS5, and signature B, which is a lot like SBS1. And then we could do the same test. which of these processes is driving the age-related accumulation of somatic mutations. And relative to the intestinal crypts, we get the opposite results. So signature A, which is a lot like SBS5, this one is doing a lot of the work. So this is all making sense to us. The somatic mutational signature associated with post-mitotic cells accumulating mutations is what's driving this age-related accumulation in neurons. And this is true in the prefrontal cortex. It's true in the dentate gyrus. It's true in our Cockayne syndrome and xeroderma pigmentosum individuals. We see this across groups, this linear accumulation. By contrast, SBS1, we don't see so much of an accumulation over time. There is evidence for SBS1 here because while those neurons aren't dividing during our lives, they do have a history of cell division. So there is the capacity for neurons to have SBS1-related somatic mutations. They're just associated with development and presumably don't change over our lives. It only goes up during our brain's development. But, when we're looking at disease, right, we kind of picked some low-hanging fruit here, right? We looked at a DNA damage-associated disease, a mutation in the DNA damage repair system. We saw more mutations. That kind of makes sense, right? That's not that surprising. So we wanted to go back again to Alzheimer's disease. And here we saw a similar phenomenon. So here we have neurons from the prefrontal cortex and the hippocampus in both control and Alzheimer's disease. And we see age-related accumulation in both. But in this red line, you could see that the Alzheimer's disease individuals have more mutations than their neurotypical counterparts. On the right side, we can see that if you normalize for age, just do a head-to-head comparison, we see on average Alzheimer's disease neurons have more mutations. And for those of you wondering what the reduction in lifespan is in Alzheimer's disease, typically there's a lot of variability to this, but Alzheimer's disease can be diagnosed. An individual lives somewhere between 3 and 12 years. There's a reduction in lifespan of somewhere in that range. And that's roughly consistent with the increase in somatic mutations that we see here in Alzheimer's disease versus age-matched control. Once again, giving us the idea that the somatic mutation rate in individual cells might have something to do with our lifespan. So we've done a couple of different things now. We've looked at DNA damage deficiency. We've looked at a neurodegenerative disease in terms of Alzheimer's. But Alzheimer's disease, we're still not quite sure for a lot of the cases what the cause is. What if we could look at something where we know the cause, a neurodegenerative disease where we understand the cause underlying the neurodegeneration? So we looked at chronic traumatic encephalopathy. Some of you may have heard of this. It gets some attention in the media. This is often diagnosed in individuals who participate in the military, individuals who play contact sports such as American football and boxing, right? Chronic traumatic encephalopathy is neurodegeneration that happens after repetitive head injury. So this allows us to do a very interesting experiment. We can look at control individuals, shown in the dark blue, and see a nice accumulation over time of somatic mutations. We could look at those individuals with CTE, shown in the red, and see, once again, more somatic mutations. But we can additionally look at an RHI group. So this is those individuals who have repetitive head injury, but were not diagnosed with CTE. So it's not just repetitive head injury that causes somatic mutations. It have to have both. You have to have the neurodegeneration and the repetitive head injury to be at a greater risk of accumulating somatic mutations. Those RHI individuals have a relatively normal amount of somatic mutations. It's only those with CTE that have more somatic mutations. So now we're really getting through a whole lot of different brain conditions that have to do with neurodegeneration, aging, DNA repair, right? And seeing that private somatic mutations can be important too. So I've told you about a few different somatic mutational processes now, right? We started talking about germline mutations when I told you about TMM161B. I told you about how we discovered this mutation by looking in the blood of the individuals. Presumably all the cells in the body have this mutation, and then for whatever reason, this mutation specifically impacts the brain. But we can also have more targeted effects. We talked about how in hemimegalencephaly, it's a developmental somatic mutation, something that impacts only part of the brain that leads to this disease. We know that because the mutation is only present in some parts of the brain, it's not in the blood, and the brain is what's affected. And finally, we talked about accumulation of private somatic mutations. So we talked about how that has to do with lifespan across lots of different mammals. We talked about how it has to do with disease as well as aging in humans. And it's not just DNA damage repair machinery that's different. It's also other neurological diseases that aren't directly associated with DNA damage repair or somatic mutational processes. So I hope I've convinced you that somatic mutations can have a big impact on our health, even if it's not cancer. And I'm going to thank my lab. This is where I learned all of the somatic mutational stuff. This is all of us celebrating all the postdocs who are moving on to bigger and better things, right? These are my funding sources up here. And I got I had the pleasure of working with a lot of these individuals. I haven't worked with all of them, but they're all first authors on the papers that I talked about today. And I'd be really excited to answer any questions that you have.
That was fantastic. If you have a question, please come up to the microphones in the middle of the room. Okay, I will start. I'm wondering how much this impacts organs or diseases that you didn't mention. For example, I study kidneys. Should I be interested in somatic mutations, or is it like, does someone look into this and there was nothing there, or is it an unexplored space for other organs?
That's a great question. So it kind of feels like sometimes in the Walsh slide, we just pick a random disease and there's going to be somatic mutations there, right? But there is a process we have for thinking about what's more likely to have a somatic mutational process as the root. The brain is an especially good candidate because all those neurons are post-mitotic. So when we have the development of the brain, if there are somatic mutations that occur, those somatic mutations persist forever, even if they don't cause a proliferative advantage. Presumably in another system where there's more turnover of the cells, if there's a damaging somatic mutation, that cell might just be replaced by something else. So in general, if we have some kind of, if we go back to some of these examples, hemimegalencephaly is, in hindsight, a very good example of something that might have a somatic mutational process associated with it because it has a defined region of the organ that's affected, and it's full of post-mitotic cells. So thinking about other organ systems, there are definitely other examples that exist. We talked about Proteus syndrome, which is theoretically happen anywhere, right? But one of the things we look for to be suspicious about somatic mutations is if a defined region of the organ is more affected than other parts of the organ. Does that answer your question? Awesome.
This is Pam. I have a question. Well, I have two questions. The first is for people with like CTE, for example, or one of these other brain conditions with more somatic variants. Can you see a signature of that in the blood at all? Or is it really specific only to, just wondering if yeah, you probably wouldn't see it in the blood, but I'm just asking. And then another question is, do you have an understanding of what the mechanism is of how increased trauma would lead to more somatic variants?
Yeah, okay. These are fantastic questions. So the first question is about whether there are also more somatic mutations in the blood of individuals with CTE. So the first thing I'm going to say is no, we didn't check, so we don't know. But we would hypothesize that there wouldn't be, because we're not just looking generally at the brain when we're looking at that somatic mutation accumulation. We're looking specifically at the neurons. So those neurons are going to be somewhat special. They're all post-mitotic. They live in the brain forever. If we, by contrast, saw mutations that we thought were happening in the glia of the brain, that might be something that we thought would also apply to other parts of the body. But in this case, we have an event that's specific to the head in terms of repetitive head injury. We're specifically seeing this mutational process in the neurons. So I would guess there probably isn't an increase in mutations in the blood, but we didn't check. The other thing is, how do we think these mutations might be happening? So there's various symptoms associated with CTE. right? You look at the brains of the individuals, you look at the pathology, we can see that there are areas of the brain that degenerate faster than others. There's a lot of impact that happens that could affect the folds of the brain specifically. There's some pathology that suggests sulci might be especially affected, for example. Ultimately, it's very difficult to tell. Model organism research on CTE is still just kind of getting rolling. It's very difficult to do a lot of this work because CTE can also only be diagnosed at death. So unlike Alzheimer's disease, we can't deal with living individuals that have the condition.
Thanks for the great talk. My question was a little bit kind of the same as the last question there, asking about the mechanism that you suspect in CTE related to, I guess, some repair processes after injury. But I guess I could continue that and say, does that questions about the mechanism say something then about the mechanism of Alzheimer's disease, of how we get these somatic mutations?
Yeah. So these are both neurodegenerative diseases, right? They both have increased somatic mutations in the neurons, right? I think that one of the things that we could say, sort of putting both studies together, is that we may have a commonality between them, that neurodegenerative diseases may have something in common. We see some somatic mutational signatures that are similar between the two. A lot of the baseline SBS5, SBS1 signatures are similar, but there are other mutational signatures that are different. So I think putting them together, I think there's still a lot more work that has to be done to get increasingly mechanistic. But I think that what I would surmise is that something that damages the DNA or the ability of a cell to repair its DNA more accurately is going to have long-lasting consequences and potentially lead to some increased death of neurons. That can't be replaced because those other cells are post my talk.
I'm wondering if you could briefly touch on the like the computational challenges in studying somatic mutations, especially when you're talking about the cells that aren't proliferating and they have like how many reads, like how do you measure that?
Yeah, no, I decided not to touch on that at all in this presentation because that could be like a whole 4-hour-long talk. Just touching on some of the challenges you have to deal with. I mentioned that, Alex Kagan had the privilege of looking at a whole group of cells that presumably all harbor a mutation. That makes your life much easier. You have many, many copies, presumably, of the mutation that exist in that sample, so you get a lot of cracks at it. By contrast, if you have to amplify the genome of just one cell, you have to be really, really confident that you're doing it right. So there's two kind of broad strategies I can tell you about for how to do a single cell whole genome sequencing experiment. One of them involves getting the amplification to be very reliable. The other one involves tagging the two different strands separately so you could check your work. If you find a mutation on one strand, you check the complementary strand and see if you agree. So in the first strategy, you try to amplify the DNA in such a way that you're not propagating error. So you look at your template DNA, your original genomic DNA, and you set up your chemistry so that you only amplify off of that template rather than amplifying off of the amplification of the previous round of pre-CR. That helps a whole lot. And you can check your work by looking at known germline mutations. So you could see, am I detecting all of the germline mutations I expect to detect? Do the mutations I see next to the germline mutations match up in terms of what I expect, in terms of heterozygous SNPs and stuff like that? The other way you think about it is strand tagging. So strand tagging means that at that first step, when you separate the strands of DNA, you tag them with a primer so that when you call every single mutation, you can then computationally check to see if on the opposite strand of DNA you agree. Lots of the mutations associated with library prep are going to be on single strands rather than both strands, especially if they're processed separately. So tagging the two different strands, call it duplex sequencing, is going to give you a really big boost in your sensitivity and accuracy.
Great. And then we have one more. We're wondering if the proliferation or turnover of the cells is going to impact how many somatic mutations it gets. I'm wondering if there's other characteristics of cells that make them more or less susceptible, maybe variation in the DNA repair mechanisms in certain cell types.
Yeah, that's a fantastic question. So it's definitely true that cells that proliferate faster are going to be more prone to accumulating somatic mutations. We see that across systems. We also see that different cell types accumulate somatic mutations at different rates. So the lowest rate of accumulation of somatic mutations in any human cell types that have been tested so far is something you might expect. It's the germ cells. sperm is the gold standard for the lowest accumulation of somatic mutations because those mutations are going to be passed on to the next generation, right? We need to keep that DNA very, very perfect if we can. Neurons have about half of the accumulation of somatic mutations over time, as do intestinal crypt cells. So different cell types definitely have a different amount of prioritization in terms of DNA repair and keeping the DNA integrity high. And that's going to depend on their function, how long-lived they are, and lots of other factors. And I think we're just getting started about like really rigorously profiling exactly which cells have what kind of capacity for DNA repair.
Questions in the room? All right, great. We'll thank our speaker one more time.